Now let's get into secondary structures. All right, the secondary building blocks of proteins. All right. So the chemical nature of these natural polypeptides or primary structures that puts constraints on the shapes that can be formed into making proteins. All right. So it's the chemical nature, the bonding, the hydrogen bonding that we'll get into here in a second. It's the bonding of these different amino acids or mineral structures. That chemical nature is what helps shape proteins. It's the chemical nature that shapes what proteins can do, the shapes that it can make, etc. Now let's truly get into the secondary building blocks of protein. So this simply means what I just said up here about the chemical nature. This simply means that proteins will have a defined secondary structure. And there are three types of secondary structures. We have beta sheets, which are these here in blue. We have alpha helix. Everybody knows about helix. Alpha helices. This is what we see here in green, as well as loops. And these loops are what connect the different types of primary structures. All right. So they connect these beta sheets to these alpha helices, and out of the N terminus or C terminus of the alpha helices, these loops can also connect to other helices or connect to other beta sheets to form different types of protein, to build different secondary structures to create quaternary and tertiary structures of, basically what we call protein. So basically quaternary structures is what is an actual full protein. It's a full structured protein. These are just building blocks. That's why they say the amino acids are the building blocks for proteins. It is the building blocks. You have to see the hierarchical level of how these proteins are being built. So this is a true functional protein. I'll give you a full example of a functional protein, a quaternary structure of a protein in hemoglobin. Now I'll get into that. So let's see how these elements come about, as well as how these elements give rise to the different types of secondary structure elements. All right. So let's get into it, family. So let's recall that proteins are polymers, which are basically groups of amino acids or mineral structures. We just went over this. Polymers are like a group of mineral structures, carbon, oxygen, hydrogen, and nitrogen. We have to truly understand how cells are formed, how proteins are formed. And we get this by understanding these words, these different words. So this is why I put these better understanding so we can truly understand what we're talking about when we're saying these words. So when we say polymers, just think of groups of amino acids. All right. And amino acids, we know mineral structures. All right. These polymers or groups of amino acids come about from the condensation reactions that are catalyzed by the translation machinery in the cell. So this simply means the water translations or hydrophobic surfaces create condensation reactions. So when we were talking about those hydrophobic residues, these hydrogen bonds are what create these groups of amino acids or these groups of mineral structures. This is how they bond themselves together. Condensation reactions, water, as you can see right here, condensation reaction binds these two amino acids together, and this is how we create secondary building blocks of proteins. All right. So I wanted y'all to truly, thoroughly understand this. It's water, the hydrophobic bond that help bring these mineral structures together, that bond these mineral structures. So we start to really see the importance of water. All right. So after that primary state, the secondary state comes, and these secondary states come by binding these two amino acids together, these two mineral structures together. It's the water. All right. So this leads to an unbranched polymer or group of amino acid residues that are linked by amide bonds. And in proteins, these amide bonds are called peptide bonds, as we've been going over for this entire masterclass so far. The hydrophobic bond, the hydrophobic bonding. All backbones of protein structures are alpha carbons, as we just went over again. All right. So carbon is more important than we have ever began to understand when it comes to life itself. All right. So no protein, what are we talking about? Proteins in plants? What are we talking about? Proteins in the human body? No protein can even be built or function properly if it doesn't have carbon. So this is why Sebi used to always say, carbon is essential. Carbon is one of the most understudied and underevaluated essential elements within the body. All right. So we got to understand carbon at the deeper level, because we talk about carbon, we talk about melanin, but nobody truly understands how these structures are operating within the body. Carbon is the basis for every living organism within this planet. Carbon, carbon, carbon. Within this planet, within our bodies, within the solar system, within the universe, it is all built off of carbon. It is that important. Carbon is very extremely important, family. All right. So I just wanted y'all to truly understand this. All right. So let's keep it moving. So now, to form two familiar regular secondary structures, such as alpha helices and beta sheets, we need several protein or mineral structure residues in a series to form themselves. For an alpha helix, we need multiple protein or mineral structure residues in a row to lie within the phi and psi regions to form its structure. So as we can see here, this is an alpha helices, and this is the backbone structure of this alpha helices. As we can see, it's looping, it's looping, it's looping. I just want y'all to see how to look at this backbone so you can truly see how this alpha helices is formed. All right. So we know that this is built off of multiple mineral structures coming together and having hydrogen bonds, hydrophobic bonding to bring these structures together. And to create this alpha helices, natural L-amino acids form right-handed helices and chiral amino acids form left-handed helices. All right. So I just wanted us to understand how these secondary structures are built. Same for beta sheets. You have to be in a series. As you can see, this is a series. And as we'll go further, there are about three different mineral structures, about 3 mineral structures per turn on this alpha helices. So we'll see 1, 2, 3. Now it gets to the next part of the alpha helices. 1, 2, 3. Now it goes to the next stage. 1, 2, 3. It's approximately 3.6. So around 3 to 4 different mineral structures that we'll see in the entire revolution of creating a secondary structure, which is an alpha helices, beta sheet, or a loop. All right, so let's keep it moving, family. So you can see that the backbone atoms can form hydrogen bonds, which we see here in blue, hydrogen bonds with their polar groups, which is the group of amino acids that I just broke down. So the carbonyl of the residue N that we see here will hydrogen bond to the amino of the residue N plus 4. So as we see, the 4 N structures being binded together once we get to those 3 to 4 residues of the amino acid of the mineral structure, the 3 to 4 residues of this mineral structure. All right, we see this highlighted here in yellow. And this is how side chains, which are illustrated here in the spheres, will then point outwards and slightly downwards on and off a helix towards its N terminus, which is the N terminus is here, the C terminus is here. So now the periodicity of an alpha helix is, like I explained earlier, and it contains 3.6 residues or mineral structures per turn. All right, and that will lead to the important patterns in the primary structure or sequence of a protein. So now we're building the foundation of the DNA within the trillions of cells that consist within our body. All right, we're understanding the foundational principles of which these alpha helix, which we know as DNA, is built upon within the trillions of cells. It's minerals, it's hydrogen, oxygen, and nitrogen that these are built upon. And some of them, of course, contain sulfur bonds, sulfur atoms as well. So we have to understand that these mineral atoms, these mineral structures are important for the structure of our DNA. It's important for the structure of our cells. All right. Minerals and water is essential for you to thrive, for your cells to thrive. All right. So this is why Sebi used to say the body contains 102 minerals. And when just one of those minerals is deficient, we are susceptible to disease. Just think if nitrogen or oxygen was missing from this mineral structure, this alpha helix would not be able to function properly to create your cells. You get what I'm saying? To create the DNA that forms the cells. All right. So we got to understand this from a whole deeper level, family. We got to understand minerals and we got to understand hydrating our bodies from a whole deeper level. All right. So let's keep it moving. Here's an example of ATP synthase, as we see right here. So on this helix, you can see the yellow residues are the hydrophobic residues, the bonding residues, and they all fall on one face of the helix. And if you count them in the primary structure, they're about 3 to 4 residues apart, as we just went over in the previous slide, because we said in most alpha helices, the periodicity of an alpha helix, it contains about 3.6 residues. All right. So same as we see here in yellow. The yellow residues are the hydrophobic residues, and they all fall on one face of the helix, which is about 3.6. And if you count them in the primary structure, as we did over here, we'll see that there are about 3.6 mineral structures per turn. All right. Per turn, full turn. All right. So we got to understand that. And that is what allows this particular helix to form a hydrophobic core. And this hydrophobic core allows this alpha helix to pack themselves against each other, to bind themselves together, basically, which of course go on to create bigger structures known as tertiary structures, or in the tertiary structures, of course, go on to build the quaternary structures, which are functioning proteins. All right. So let's keep it moving, family. So as we keep going, you're going to see even more and more and more and more the importance of water, the importance of minerals. All right. So let's keep this moving. Now, let's turn to beta sheets. Multiple residues in a series can form a beta sheet. You need multiple beta strands to hydrogen bond together to form a sheet. All right. Here is a 4-stranded beta sheet in cartoon representation. Each strand is illustrated by an arrow going from its end to the C terminus, as you can see here. So sheets can contain parallel strands, anti-parallel strands, or a mixture of both, as we see here. Parallel strands, anti-parallel strands, and sometimes we see both. As you can see here, this beta sheet is mixed. Anti-parallel strands often come from the adjacent parts of the primary structure, and then they would be linked by a short loop. All right, and we're going to get into loops here soon. These loops help link these beta strands to become beta sheets. So once these loops connect all of these beta strands can become a beta sheet. All right, so I want y'all to understand that. All right, so let's look at the backbone structure of beta strands. All right. So in a single beta strand, the backbone structure is an extended conformation, meaning it's an extended polymer or group of mineral structures within a linear form, which they call extended conformations. All right. N terminus to C terminus. And the hydrogen bonding of the backbone amino acids and carbonyl groups between adjacent strands is what forms a beta sheet, as I just explained. So we see here the extended conformations, or the extended bonding of these mineral structures. They are now being bonded together, as we see here on the dotted lines. They are now being hydrogen bonded together to create a beta sheet. All right. So we have to understand this, because this is a important level of the secondary structure of building proteins. We got alpha helices, we got beta sheets, and we have to understand beta strands to create the beta sheets. And then we also have the loops that connect them. All right, so we're going to get into loops here in a second, but I wanted y'all to understand this is the backbone structure of what a beta strand looks like. This is the backbone structure of what this true beta strand looks like. All right, still built off of the CHO arrangement family. This is the second level. So all of this is still being built off of these mineral structures. All right. Nobody has broken this information down like this. I ain't seen this nowhere. And you probably won't see this anywhere, because I had to do thorough studying, family. You got to truly study the body. You got to truly understand biochemistry. You can't just say all this mumbo jumbo that they're giving. You got to truly understand what you're talking about. You got to understand what you're talking about. All right. So now let's get into the loops. Loops and turns will connect the adjacent secondary mineral structures and elements. Our first example here is 2 strands of an anti-parallel beta sheet, which are connected by a small loop. Then the second example is a helix followed by a loop, followed by a strand. All right. So as you can see, these loops is what helps connect these different types of secondary structures together to then form motifs, which go into building domains, which are tertiary structures and then quaternary structures. I got to keep saying that so you can keep understanding the hierarchy of how proteins are being created. All right. So most loops will fall at the surface of a protein, the surfaces of proteins, and contain a lot of polar and small residues. So this has a lot of small polar residues, which actually allows it to connect these alpha helices and beta sheets, or beta sheets to beta sheets. All right. These loops will also form the shapes of the active site because they have a lot of conformational flexibility to really shape themselves around the ligands. So this simply means in proteins, there is an active component within basically the core of what they're calling the active site of a protein. These loops is what actually help hold these active sites together. And this is a lot of times where these loops get its information from, and to be able to help build the rest of the structure of the protein to do whatever a protein is needing to do within the body. All right. Such as hemoglobin. I'm gonna keep saying hemoglobin because that's one of the examples that I gave in this masterclass at the end that we'll see. But hemoglobin basically sends the oxygen to whatever parts of the body that we need. It goes and grabs the oxygen from the lungs, takes it down into the blood, takes it down into the cells, as well as hemoglobin also takes acidity, dissolved carbon dioxide, which is deterioration. Basically, it takes that carbon dioxide from the tissues, from the cells, from the blood, back to the lungs so you can breathe it out. So you're breathing in oxygen and you're breathing out carbon dioxide. All right. So this is what keeps the body healthy. And the active sites within hemoglobin is iron. So I just want y'all to understand how loops are created and all of their conformational flexibility in holding those active sites together, holding that iron within the hemoglobin as it's transporting that oxygen. You know what I'm saying? I just want y'all to truly understand the full aspect of what's being said here and how our body is operating. We need to know how our body operates to truly understand it. All right. And this was our ancient information. We've been through this stuff. You know, we had to go through a period, but now it's time to re-educate ourselves on our level. But we have to truly understand the full granular scale. We got to go to the intercellular scale. We got to go to the protein, the amino acid, the mineral structure to understand ourselves on the deeper level. All right. So let's keep it moving. Now that we understand loops, we understand beta strands, how they're formed, understand beta sheets, and we also understand alpha helices. So let's wrap up the secondary building blocks by looking at alcohol dehydrogenase, an enzyme important in energy metabolism. All right. Metabolizing energy. So when we eat food, catabolism, anabolism, which I broke down on Instagram, go back and look at that video if you haven't seen it. It's a picture talking about metabolism, explaining the true understanding of metabolism. So looking at alcohol dehydrogenase, it contains a mixture of alpha helices, beta sheets, and loops. Can we start to imagine how much minerals one needs for these biochemicals to be sufficiently produced? Can we start to begin to understand how much minerals and water is essential and necessary for these biochemicals that are happening within the body to sufficiently be produced? Can we begin to start seeing it? Can we begin to understand why we need minerals in abundance? All right. So we can start to see it, because all of these secondary building blocks that have now come together, that have joined together due to these loops, beta sheets, and alpha helices, we already know that those are built off of the CHO arrangement. So we can see that when they come together, we understand why the body needs so much minerals, because everything within the body is built off of these minerals. I'm just taking you to the intercellular level. I'm taking you to the inner protein level, the inner mineral structural level, to be able to truly understand why the body needs so much minerals. All right. So let's keep this pushing, family. So now we've seen how backbones of a protein can be arranged, which are the alpha helices, beta sheets, and loops. This level of organization is the secondary structure. All right, so now let's get into tertiary and quaternary.
If you enjoyed this video, it's a probable guarantee that you're gonna like this one as well. So go ahead and give it a gander, and remember to keep it cosmic.